Okay, welcome students. Welcome back again. So we are going to deal with the next topic, that is a plant growth and growth regulators. So what actually the growth is, the plant growth? It is a growth. It is an essential characters of all living organisms. Then we can say development of an entire organism from a single cell, or in the higher plants, we can say development of a mature plant from a tiny seed is said to be a growth. So there is a increase in size, shape, weight, height, length, and biomass. So overall biomass due to the growth only. So it is a permanent and irreversible change in an organism. Okay, directly we can say this. So the growth occurs due to the cell enlargement and cell divisions. Okay, so it involves various physiological and we can say biochemical processes. So it also needs a favorable external and internal environmental conditions also. So we can say basically according to the definitions, it is an irreversible permanent increase in size, volume, or mass of a cell or organs or the whole organisms. So this is a proper definition we can say. So there are phases of growth, various. There are three phases, major phases of growth. So in plants, growth is confirmed with only two meristem, only to the meristem we can say. So it takes place at the apices of root and the stem. So these growing regions are called as apical meristems or the primary meristems. Okay, so the meristematic cells have to pass through uh, we will say three phases. So they are the first, that is a cell formation, then cell enlargement, and the third is the cell differentiation phase. So the one by one we'll take. That is the first, that is the cell formation phase, or we can say cell division. So it is the first phase of growth, so in which cell division and new cells is formed. So cells are small in size, having active nucleus and cytoplasms. So the cell wall are thin and contain large quantity of solutes. Again, the cells are ready to absorb water, food, and have a high metabolic rate. So cell divide and redivide to form new cells continuously. So again, next step, that is the cell enlargement is formed, that is cell elongation is formed. Okay, so in this stage cells are now enlarged in size, becomes enlarged in size, absorbs large quantity of water, so osmotic pressure is increased, high turgor pressure is formed. So this turgor pressure stretches the walls of growing cells. So a large vacuole appears in the center, and we can say protoplasm passes towards periphery. So it leads to the increase in the we'll say length and size of the cells. So then it forms we'll say the cell maturation phase or the cell differentiation. Differentiation is been given. So it is the third phase of growth, is called as the cell maturation phase or the cell differentiation phase. So the metabolic activity of cell increases and the cell accumulate food material in these stages. So the cell mature and it differs into various shapes and sizes. Thus the cell becomes specialized to perform specific functions, we can say in the plant body. So and the cell wall becomes thickened, okay, and the cell organs organized into tissues and then into organs. Okay, so these are the three phases of a growth. Now the next, that is we can say the grand period of growth. So basically, so this term was usually uh, was first time used by the German physiologist Sachs in 1873. So according to him, the total period required by an organism to grow and to and to acquire a permanent form is known as a grand period of growth. So, so the rate of growth is not uniform, all times during the period of growth. Okay, basically. So it includes, it's divided into three, we can say, stages. The first is the initial stage, that is known as a lag phase. Then the second, that is a middle stage, that is a log phase. And the third, that is a third stage, that is a steady, or we can say maturity stage one. So the first, that is a lag phase or initial stage. So it is in this cell formation phase is formed, where growth increases slowly. Okay, the starting point. And the next thing, in the log phase, what happens, it is the cell elongation phase. Okay, where growth is very rapidly, it happens in log phase. And the third stage, we can say, or the steady phase or the mature stage, it is the cell maturation stage, we can say, where growth becomes slow after these stages. So in the so this, it is represented in the graph, we can say this. So so in the graph, growth rate against time shows the growth curve, that is a sigmoid, or we can say S-shaped growth curve. So the sigmoid curve represents integrated sum of the curves for each growing organs and the cell. Okay, so this is a grand period of growth. Now, what are the factors affecting? The various factors are there which are affecting the growth. So we can divide it into two part, that is the abiotic and the biotic one. So the first, that is the abiotic factors or the non-living factors. So the first, that is a food supply. So the growth is in the food supply. So growth is directly proportional to the food supply. So if the growing regions do not get adequate food supply, so the growth slow down and ultimately stops. Okay, so the natural process, we can say. Then the next, that is the nutrition supply. The same thing again can happen. So plant needs several major and the minor minerals, so you can say, which are essential elements for normal growth and development. So these nutrients come from the soil and are being used in various metabolic activities. So the growth, we can say, increase when the nutrient supply becomes limiting. Okay, so it is one of the factors. Then the next, that is a soil one. So this is also called as we can say edific factors of the plant growth. So the soil which has a good moisture capacity, that is we can say loamy soil, and aeration causes a good growth. Okay, so the adequately humid soil with the right balance of all the minerals and nutrition is one of the essential factors that directly affect the plant growth. So the right pH balance, that is adequate alkalines or the acidity of the soil, is also important for the growth of the plant. Then the next, we can say, that is the water, which is again very important factor. Water acts as a medium for all metabolic and physiological processes, so it is one of the very needy one, and it is essential factors. So next, that is oxygen. So it is again necessary for in the plant for the cellular respiration, in which food minerals are broken down and energy is released in the form of ATP molecules. So this energy is used for various activities of the cell and is directly involved in the growth and the development process. Next, that is the temperature again. So the variation of temperature of the soil is very we can say, which in which plants are growing also affect the plant growth. So the most plant species prefer temperature in the range of 20 to 40 degrees Celsius. So the higher soil temperature results in heating of the soil solution, which is determined for normal functioning of the root cells. So it also results in the rapid, we can say, evaporational loss of water from the soil. So the low temperature causes chilling, or we can say cold injury, while temperature above 45 degree causes denaturation of enzymes, heat injury of to the plants. Okay, so the optimum range of the temperature should be the 25 to 35 degrees Celsius for maximum photosynthesis and respiration, differ from species to species. Okay, then the next, that is a light one. Again, light is very important for the plant growth one. Okay, so basically we can say the light influence many physiological and growth process of the plant. So plants which grow well under bright direct sunlight but show poor growth in shady conditions are called as a photophilic plants, whereas the plants growing better under low light conditions are called as a photophobic plants. So the lights can affect morphologically of the leaf and the stems of the plants. Okay, variations in the intensity of quality and the duration of light variously affects physiological process such as seeds, germinations, and flowering in different plants. So the effect of light on reproductive growth is it's also found reproductive growth and which is known as a photoperiodism. Okay, the various colors of lights are there, which its functions are different one. Okay, and the last one is that is the plant growth regulators one. So in this organic substance, uh, so it is an organic substance produced within a plant and which at a low concentration promotes or inhibits or modify the growth process. So this the effect is usually observed at the site other than the phase of synthesis. So this is the abiotic factors of, or we can say non-living factors for the growth. And what are the biotic factors? That is the living factors. The first one, that is the microbial population. So the biotic factors refer to the living, we can say macro and the microorganisms. So these living factors may improve or retard the plant growth depending on we can say how they interact with the plant. So the disease causing fungi, bacteria and viruses are we can say abiotic factors affecting the plant growth. Okay, so the next, that is other biological conditions are there. So the presence of weeds and the allelopathic chemicals. So allelopathic, it is a process, or we can say the plants which directly or indirectly affects the nearby plants. So you know, some chemicals are present. For example, we can say Congress weed. Okay, so the, you know, the chemical allelopathic chemicals in the soil also affect the plant growth. So weed populations less in the growth because of the exclusion of nutrition from the soil. So characters of seeds are there, proper orientation of embryo, then during the seed germination proper enzymes activity also affect the plant growth. So these are the some factors, biotic and abiotic factors affecting the growth. Now the next, that is the plant growth regulators. So what are the plant growth regulators? So the phytohormones or the growth regulators are we can say organic substance naturally producing in plants which controls the growth or other physiological functions at a site remote from its place of production and acting in extremely minute quantities. So this is the definition, proper definition. Next is the plant growth regulators are substances that are synthesized in a particular cell and transferred to other cells where an extremely small quantity influence development process. So basically the plant growth regulators are the, we can say, naturally or chemical process which helps to plants to we can say inhibit or promote the plants for betterness of the plant. Now, so what are plant hormones? It is basically divided into again three parts. Okay, so various chemicals are there. Okay, growth hormones are there, various growth regulators we will say. So growth promoters are there, then the growth inhibitors are there, then the growth promoters and the inhibitors both are present. So we have to select the which chemicals we have to use. The first, that is a growth promoters, that is auxin, gibberellin, cytokinins. Then the growth inhibitors, which act only the growth inhibitors. आपल्याला जर पाहिलं growth inhibit करायचं, then abscisic acid is needed. And if we need both quantity, both quality, then ethylene is there. Okay, it works as a promoter as well as inhibitor also. Now one by one we'll take. It is a basically a comparative chart of the plant growth regulators. So the first part, that is auxin for the stem, we can say It promotes a cell enlargement behind apex and promotes cell division in the cambium of the stems. Okay. And again in the gibberellins, it promotes cell enlargement in the presence of auxin also. With the auxin we can use it, and the promote leaf growth also. Okay. So next, cytokinase. It promotes cell division in apical mesostems and cambiums too, together. Okay, it promotes leaf growth also. The same thing which is found in the gibberellins. Then abscisic, it inhibits growth during the stress. Example, drop salinity, okay, water logging, and the same thing is found in the ethylene also. Okay, it's inhibit the growth one. Okay, for the stem, and for the root, next part, for the auxin, in low concentration promotes growth, so in high concentration inhibits growth. Okay, so both the conditions form. So the promoters growth from the cutting and calluses. Okay, and then in gibberellins no such characters are found. Again in gibberellin and abscisic no such type of characters are found, whereas in the cytokinase inhibits primary root growth are found and promotes lateral root growth is found. Laterals branches we can say. And in ethylene also, in the root it inhibits the root growth. Okay, now the next again, further we study the floral buds. Okay, so in auxin and in gibberellins, and again you can say in all the conditions, cytokinin, abscisic acid, it promotes bud initiations, wherein cytokinase promotes lateral bud growth also and breaks bud dormancy also. Okay, whereas in the flowering, during the flowering conditions, uh, there is no any action is found in auxin and the cytokinase. They don't have any action taken. So whereas in the gibberellins, abscisic acids, we can see that it promotes in long day plants. Okay, inhibits in short day plants and acts as a substitute for red light, and again antagonistics to the abscisic acids. Okay, and then whereas in the abscisic acid, it promotes short day plants, inhibits long day plants, acts as a substitute for red light antagonistics in the gibberellins, and in the ethylene it promotes in the pineapples. Okay, so these are the some, we can say, very normally I have studied this comparative chart for the plant growth regulators. So these are the 5 growth regulators. So this comparative chart can help you to understand very well. And it is again further, I have given some applications which can again help you. So root in for the applications of auxin, root initiation is there, induction of flowering is there, fruit setting, then parthenocarpics, prevention of premature drop of fruits, then seed germination and dormancy, apical dormancy is that, weight control, tissue and organ culture. So these are the applications of auxins. In the exams they can ask you. So these next application, that is a cytokinase, cell division, then tissue differentiations. Okay, senescence is that, dormancy is that, the protein synthesis is there, stomatal movement is that, morphogenesis, again gibberellins. All these are conditions are been applications are be given, you can apply it. Okay, you can use it for the ethylene applications, for the ethylene ripening of fruits, changes in sex expression in flowers. Okay, and the last bit is the abscisic acid. Thank you very much.